Hi everyone, I'm going to walk you through a little bit of tissue culture that I'm doing today. Hopefully this will be interesting. But first thing we're going to do is we're going to collect the media, and you can see that there's all kinds of different medias in here. Each one of these medias is basically for a different set of cells. And these have all the amino acids, nutrients, glucose, growth factors, all kinds of things that cells need. And the reason there's so many different types is because different cells would grow in different places in the body and therefore have different environments that they grow in and different combinations of nutrients and so forth. So these things are pretty important along the way. All I'm doing today is I've created some media for some fibroblasts, some primary human fibroblast cells. So we're going to warm those up. We're going to warm them up along with some other reagents that I need to split the cells. And we need to warm them up because these cells are at 37 degrees Celsius, that's body temperature, and so if we put refrigerated cold media right on them, it would be like you jumping into an ice bath, but without any protection. These are just the cells, so we need to warm them up to body temperature, and then they'll do fine. Here we have our incubators, our tissue culture incubators, and these incubators are hold 37 degrees, but they also hold 5% CO2, and that 5% CO2 is really important because it seems pretty high. Natural air is actually usually less than 0.5%, certainly less than 1 or 2% CO2. But in our cells, where our cells are doing cellular respiration and producing CO2 in our body, the cells are naturally in about a 5% CO2 level. And that's important because that's why CO2 diffuses away from our cells into our bloodstream and out of our body. If you ever watched Apollo 13, one of the things they were really worried about is if the CO2 in the capsule got over 5 or 6%, because if the natural air outside gets over 6%, your body quits diffusing the CO2 from the cells out into the air, and actually you can die of CO2 poisoning. But the cells themselves are at 5% CO2, and so that's where our cells grow. We have cells growing in these little flasks here. You can see the media at the bottom. The cells will be at the bottom, and we will look at them under a scope in just a second. Alright, so now you can see that I have put the cells on our phase contrast scope. And this phase contrast scope is actually needed to be able to see live cells, because live cells are pretty clear. That's why normally when you're looking at cells, you're looking at cells on a slide that's been stained, or you have to do some fluorescence imaging with immunohistochemistry like you guys learned about last week. So you have to find a way to create the contrast in the cell. But the phase contrast basically takes advantage of the fact that there are different densities inside the cell. And just like light refracts when it goes from air to water, like something looks like it's at a different place in a swimming pool, for example, that same light is going to refract as it goes through different parts of the cell. And a phase contrast basically just amplifies that so you can see that contrast of the cells. And so we can see here on the screen, hopefully, we can see that there are our cells. Those are the cells that are at the bottom of that flask. These are fibroblast cells that are growing. And you can also see there that they are relatively confluent. There's still space for them to grow, but I need to propagate these and make a lot of them because we are using these to study HPV. These are the types of cells that would be infected by HPV, and we are using some of the oncogenes from HPV to try and study how HPV causes cancer, like cervical cancer and so forth. And so today I will be spreading these cells from this small flask to a much larger flask so that I can get more cells growing to use. And that's what we will come back and do. In the meantime, we are just waiting for our media to warm up and be ready to go.